Good afternoon, everybody. It's my pleasure to welcome you at this webinar of the OncNGS project that Cienzano leads within the framework still of the Horizon 2020 program. It's a European Commission project. My name is Mark van den Bulke. I'm the head of the Cancer Center at Cienzano. We have 4 presentations that we will present one after another. 2 are recorded, but myself and Rosanna Alessandrello will present live. Just an overview of what we're going through. So I will give a general introduction to what we call the market engagement for our OncNGS pre-commercial procurement. As you all know, probably, or most of you know, we had launched the tender already beginning December last year, but due to too few bids, we have been obliged to relaunch, which is now the preparation of that. And during this session, we would like to, let's say, go back over what is OncNGS and give you some key information, and then indicate a little bit what is particularly changed with respect to the previous tender. I will give, as I said, a brief introduction. Luca Mazzarella from the European Institute of Oncology in Milan will present the clinical and patient interests, and Pierre Sujobert from the Hospice Clinique in Lyon will give more details on the technical challenges. And then Rosanna Alessandrella from Aquas, she will present the OncNGS PCP process, which I must admit is complex and is still, we're still learning ourselves how it really works. We hope to get also some feedback from the market. You can send us questions to the chat. You can also, let's say, contact us through the website. You can make suggestions through the website. And at the end of the webinar, if there is still time, we will open a session for questions. The next slide. This is just to, let's say, indicate that cancer is a major public health challenge in Europe. It will not decrease, it will increase probably due to the aging population. We will have higher cancer incidence in the coming decennia. And the economic impact of the disease is estimated for Europe on about 100 billion euro annually. As all of you are probably aware, cancer is very high on the agenda for the European Commission. They launched the European Beating Cancer Plan. It's in full execution through the Europe for Health program. There is the mission on cancer, and it indicates that the European Commission sees it as a holistic strategy to reduce cancer burden, reduce inequalities within Europe on cancer care and control, and the European Commission puts it as a very highest importance for them. The next slide. Cancer is OncNGS is in fact focusing on providing a solution for patients with an advanced cancer, where standard practices are difficult or not possible anymore, or have very little benefit. And the treatments in such case often benefit from a more tailored, stratified approach. They call it, or we call it, personalized or precision medicine approach. And the final aim of such a stratification is to more accurately define which patients benefit from a particular treatment and who is not benefiting. And so, let's say, eliminate unnecessary treatments for patients, and definitely it could reduce the burden of unwarranted treatments and favor in general the quality of life of the patient. The next slide. The challenge that we wish to address in this PCP as buyers to the market is providing us with an efficient molecular RNA DNA profiling approach on tumor-derived material in liquid biopsies by means of a pan-cancer tumor marker NGS-based analysis, and integrate that with an ICT decision support system, including test interpretation and reporting. Now we are 8 buyers coming from 5 countries in this PCP. So we're from Belgium, France, Italy, Spain, and Germany. Now, as an example, the next slide of a particular application, I will not go into very detailed, but it's very often that we would like to help clinician in giving a more, let's say, founded decision on treatment, and OncNGS would enable to create the possibility for clinicians to somehow match their patients with other patients that have a similar profile as resulting from the OncNGS analysis. But of course, the profile itself is not all the information. You have to combine it with other clinical information. But the treating clinician could get a better view on what treatment is best suited for his patient. And on the next slide, based on these elements, also it can be envisaged to enroll patients in particular clinical trials. And in fact, OncNGS should also enable or facilitate the installation and the setup of such European trials, and what would strongly favor the evaluation of the efficacy and effectiveness of treatments due to the increased statistical power that you would obtain when you have larger trials. The next slide. Some general information on the project. So the overall aim is to develop an integrated solution for testing, analyzing, reporting, and storage of next generation sequencing medical data within routine healthcare diagnostics. But of course, it could also be used in research applications. There's about 12 million euro foreseen, of which 70% goes to the PCP. And if you want to get some more information from the site of the Commission, you can look in the Cordis website or on our website where you can find more information. I must say that the website is being updated now, but by the end of this month, well, let's say by the end of next week, we will have fully updated the website with all the information pertaining to this new relaunch. The next slide is giving you an overview of who are the buyers. From Belgium, we have Cienzano and the Institut Jules Bordet. From Italy, we have several partners linked within the Alleanza contro il Cancero. From Germany, we have people from the Charité and from the Ludwig Maximilian University Hospital. From Spain, there is people from the Catalan Institute of Oncology, and from France, we have the Institut Curie and the Hospice Clinique de Lyon. But next to the buyers, we also have in the next slide a group of supporting entities. So we have Aquas, who is an expert body in PCPs and in innovation introduction in Catalonia. We have IPSAL and VIO, 2 research institutes that help us with more giving their insights from the, let's say, scientific point of view into the precision medicine implementation. L'Institut National de Cancer, who is a focal point in France for anything that deals with cancer. We have the Clercam Partners, who is our support to everything dealing with IP and regulatory. And we have somebody from the Cancer Register who can help us with thinking on what should be reported that is useful for the cancer registers in Europe. The next slide, just a list of all the members in the consortium. And the next slide, just a simple indication how the process is. We should start with selecting 4 bits to enter in the phase 1, we called, where you design your solution. Then in a phase 2, 3 will be demonstrating the analytical performance of the solution. And then in the final phase, we hope to have 2 that will be able to demonstrate in phase 3 the clinical performance assessment. So that's in a very schematic way. Rosanna will indicate in her presentation a little bit more on the details of the phases and the budgets linked to it. What we hope to obtain from OncNGS is definitely that in the European Union we get more standardization and harmonization, so somehow come to common guidelines. Already within the discussions of developing the tender, there was a lot of issues on data sharing, which is essential for making this happen in a European context. We also, many of us are involved in the discussion on the European health data space, so this project will definitely be integrated into that context too. We hope to stimulate cross-border purchases, which may reduce the cost for the healthcare systems. There is a vast interest in discussing among the experts through the e-consults. I think the COVID has stimulated teleconsultation and telemonitoring, everything that goes with digital. One of the things that definitely is interesting is molecular tumor boards. We hope that it stimulates multicentric clinical trials. And in the end, as I indicated and as an example, this patient matching tool is certainly something that from the clinical side is something that there is a lot of interest in. An idea from the market side, with Belgium, I say here 11 million, but I understood from the last StatBel that we are more close to 12 million. We have an incidence rate of about 70,000 a year. In Belgium, the OncNGS would probably integrate it in the standard reimbursement scheme by our sickness insurance, RISIF INAMI, NIHDI in English. And we estimate today we have about 12,000 tests that are within the scope of what OncNGS could be used. Although we are now focusing mainly on tumor biopsy material. The reimbursements is at 350 euro, but that's for smaller panels. So that's in this case, if the panels get larger, the reimbursement would probably be considerably higher. There is no agreement today for most of the liquid biopsy applications, but they're under discussion. And we expect for Belgium somewhere 10,000 tests a year. So if you enlarge that to the scope of what the buyers countries are covering, it would be between 100,000 to 200 tests a year. The next slide is giving you some idea on the market. It's estimated the whole market at 120 billion euro per year by the US and the EU5. In Japan and China, if you include it, it's another 20 billion by 22. And the market of next generation sequencing, and I think this is already all data, was estimated at about 260 million, but I also saw numbers that reached up to 1 billion. So it's really a huge market that there is open for an application just as OncNGS or the whole NGS business. Some general lines, we will give you more details in on a specific set, but the general lines of thinking for, let's say, the changes that we applied in the OncNGS call for tender now is to remain very close to what we originally wanted, but a number of the must-haves that we had in the first tender have become nice to have. So we increased the flexibility. Also in the way the coverage of the gene panel markers that we find important, we also increased flexibility how to address that. And then also on how the integration of the interpretation algorithms of the data, how they are integrated into the solution, into the workflow. For example, through cloud services, we're open for propositions there too. An additional thing is that we understood that some people had troubles in getting the response, the proposal ready in the period, in the 2 months period. So we extended that to 3 months. Now, finally, we summarized a little bit in 2 slides what today is, what now is, and what OncNGS would become. I think now we have solutions probably matching closely to what OncNGS wants in different elements for tissues. The market on liquid biopsy is very rapidly growing, and there also some elements are already present. But in fact, what we would like to do in OncNGS is to bring that all together in an integrated way. And we understand that this is a challenge, but I'm sure that together with the buyers and some of you suppliers, we can bring out a very valuable project at the end. And in the next slides, and that's my last slide, yeah, it's a little bit small. What we also wanted to indicate that the very important element of OncNGS is that it has to be integrated into the clinical flow, and the current practice in oncology diagnostics is something that is run in the premises of the hospitals. So it should be something that is definitely implementable somehow in hospitals or universities, whatever, but not in a centrally driven system. So it's more a federated system that we envisage. Now, I think I want to end here. I thank you for your attention. And Barthélemy, I think we can now move to, I understood you will upload the two presentations, the one from Luca Mazzarella and the one from Pierre Joubert at once. And it will be about 40 minutes for these two presentations together on what is interesting for from the clinical side and some technical insights.
Okay, so good evening, everyone. I'm Luca Mazzarella. I'm an MD PhD. I'm the head of the Translational Oncology Lab at the European Institute of Oncology in Milan and the coordinator of the genomics working group of the Alleanza contro il Cancro consortium of 26 cancer-oriented hospitals in Italy. My point today is going to be to illustrate the advantages and ongoing issues in the implementation of liquid biopsies. And these are briefly listed in the slide that I am showing here. And we will now briefly go through each of these points. So for sure, I will start by pointing out that liquid biopsies are increasingly there is mounting evidence on how this technology can really provide precious information for the identification of patients that are that maintain minimal residual disease after macroscopically radical surgery. And this has actually been demonstrated in a number of trials in various diseases. Here I'm listing trials showing the recurrence-free survival in major diseases like breast, lung cancer, and colorectal cancer, where you can clearly see that the almost 100% of the patients that show persistent minimal residual disease detected by liquid biopsy after surgery relapse within the follow-up time frame, whereas hardly anyone with a negative circulating tumor DNA will relapse over the months and years to come. So this is clearly likely to greatly overperform any predictive model that is currently in place for the prediction of the risk of relapse. In the metastatic setting, it is also proving of great advantage, and it is actually an established technology for decision making, especially in the context of lung cancer, where the liquid biopsy for the T790M mutation of EGFR is now an approved biomarker for switching to last generation tyrosine kinase inhibitors. But beyond these indications, also additional types of genetic alterations, like, for instance, non-single nucleotide variations like copy number alterations or splicing isoforms, etc., are being used to monitor the disease response to treatment in the metastatic setting, and they represent certainly an area of valuable improvement in the future. Besides these, if you want, technical advantages over established technologies, there is, of course, as we all know, one major factor that is key in the evaluation of liquid biopsies nowadays, and is the non-invasiveness of the technology. Clearly, this is a technology that has advantages for the patients, and this, beyond being obvious, is now also evidence-based, as this little but interesting study from the Princess Margaret shows. A vast majority of the patients, of course, at this institution preferred the blood tests over tissue biopsy, as you see in this table, but they were even agreeing to trade some sensitivity in the test in order to have minimal invasiveness, and this is highlighted by the 6.2% difference that was deemed tolerated before switching to the liquid biopsy from the solid biopsy. But beyond these two major advantages, so the non-invasiveness and the superior sensitivity in some specific settings, there are a number of issues, and in my personal opinion, most of the issues they boil down to one specific point, the issue of size. How big should a liquid biopsy panel be? Of course, we can opt for a small panel that only looks at few alterations. This is quick, it's simple, and inexpensive, but of course, this has very limited applicability, and this is, of course, the case, for instance, of the EGFR T790M alteration. Of course, a large liquid biopsy panel would be much more versatile. It could be used for many more clinical indications. But at the moment, these panels are slow, complex, and expensive, and they can only be implemented in some specific settings. And these may also be suitable for different types of alterations that are non-canonical for liquid biopsy, like copy number variation, the additional issues on liquid biopsy, they boil down to this particular problem. But let's look at the other ones. Turnaround time. So turnaround time at the moment is a key determinant for for instance, the enrollment of patients in clinical trials, as you can see in the graph on the right that analyzed the reasons for failed enrollment in a molecular tumor board in the States, where one of the key issues for non-enrolling patients with a positive indication for targeted therapy was the time to decision that was heavily determined by the time it took for the genetic information to be obtained. And a recent small study presented at the World Cancer Lung Conference last year showed that there is clearly a halving of the time that it takes to obtain the results when you're implementing a liquid biopsy-based genetic testing in non-small cell lung cancer, as opposed to solid biopsy. And of course, this at the moment is an issue, because if you want to carry out an analysis with a large panel, you have to send your sample off to a centralized laboratory, with all the turnaround time issue that this may pose. But of course, as we will see in the last slide, a different model for the implementation of liquid biopsy may contribute to reducing this key point. And in this slide, we also see data from a recent trial in gastric cancer, where you clearly see that two trials with very similar indications, in which in one case, in the GI screen, the enrollment was based on tissue biopsy, whereas in the Godzilla trial, enrollment was based on ctDNA, the time to enrollment was reduced from 35 days to 10 days. And as you can see in the graph on the right, you clearly see that the red line, which is the enrollment from the moment Godzilla was launched, skyrocketed the moment the ctDNA based trial was started. Cost then is the other issue, and this again has to do with the size of the panel that we want to implement. This is a recent analysis carried out by an Italian group in which they explored different models for managing patients in the first and second line of non-small cell lung cancer, in which they explored the health economics aspects of a completely tissue-based strategy, which is the more orthodox strategy being implemented nowadays, a mixed strategy that is also what has been happening in the last couple of years, where you use liquid biopsy in the first and second line, and a potential strategy in which you try to base all possible decisions firstly on liquid biopsy and only if necessary on tissue biopsy. And to cut the story short, the message was that the combined strategy and the potential strategy, so both strategies based on liquid biopsy, were significantly more cost effective. But, and this is a major issue in the point, this was only if the cost of the liquid biopsy was kept at the current standards for a single gene cost, 250 euros more or less. Of course, with liquid biopsy panels where the size and the cost is considerably higher, this is certainly not the case. And this directly poses an issue of equality of access. There are very few studies on this point. There is one recent study that was conducted in Colombia that, of course, may not be considered a representative nation for healthcare systems like those in Europe, but at the same time, the considerations are in Europe are very similar to the ones posed in this particular paper, in which in the end the use of HER2 monitoring using liquid biopsy in the management of HER2 positive breast cancer was considered basically not effective for the Colombian healthcare system and not applicable. So therefore, if we want to have a large-scale implementation of liquid biopsy, the reduction in cost is an imperative. Otherwise, this will be limited to very specific context of very specific patient population with a very low equality of access. Another issue that is common with solid tumor testing is the issue of the standardization of the reports. So the amount of information that is collected in these studies is very high, and this paper from the American College of Pathologists a few years ago compared the agreement on the types of information that would be reported in a typical report from 44 molecular pathology labs in the States. And as you can see, the level of heterogeneity for different categories of information was very, very high. Lastly, we really think that a more widespread implementation of liquid biopsy, especially in the metastatic setting, may really be key to increasing the access of patients to clinical trials with targeted therapy. There's a number of designs that can be foreseen where you use liquid biopsy. You can use it to pre-screen, you can use to screen, you can use to monitor the disease, etc., etc. But importantly, there already is evidence that when you provide clinical recommendations based solely on liquid biopsy, either because there is no information coming from the solid biopsy, or because there is even disagreement between the solid biopsy and the liquid biopsy, maybe due to clonal heterogeneity of the tumor, then in a vast majority of the cases, you do provide information that is valuable from the clinical standpoint, because as this very nice study on JAMA Oncology showed, you can induce responses, meaningful responses, when you're basing your treatment solely on the liquid biopsy. And finally, a major issue is the model of implementation of liquid biopsy. As we briefly said at the beginning, at the moment, you can easily implement a small-sized liquid biopsy panels locally, so every lab can run a droplet digital PCR on EGFR T790M, for instance. But then larger panels that are based on NGS are at the moment are mostly implemented centrally by large companies that, of course, aim to rationalize the resources and to obtain, to increase the database of background information to provide more accurate results. However, this is a model that, of course, is going to be optimal in the initial phases of the worldwide implementation of liquid biopsy. In the present context where demand is growing and it's probably not going to be met in the best possible way by an entirely centralized model, this model should probably change. And even then, this model poses major issues on privacy and accountability, because there is, of course, a data transfer, especially in an ecosystem like the European one. With the advent of the GDPR, this may be a major issue, a major obstacle to the implementation of large-scale liquid biopsy. This, of course, poses logistics problems. You have to organize the shipment of the samples, and the samples may degrade, then may be lost. There may be a number of problems. And of course, this is going to increase turnaround time. Of course, for low volume centers, this is still the centralized model is still a preferred option. But for large volumes, for large volume centers that can sustain a certain volume of analysis, then turnaround time is likely to be reduced if the analysis is carried out internally. Of course, when we are talking about decentralization, doesn't necessarily mean that every single hospital will need to carry out their own testing. It may also mean the transition towards a hybrid, if you want, system where a set of hubs is defined as the centers where the assay can be run for a number of spoke hospitals in the network. So in conclusion, what we think is that the liquid biopsy-based disease assessment in principle is superior to tissue-based biopsy for a number of reasons, and therefore it's highly desirable. It's better tolerated by patients, it's more accurate in detecting MRD, it's very highly sensitive in monitoring metastatic disease, may be more sensitive in identifying resistance, and it may be also very useful in allocating patients, potentially even more effective than current methods. However, there remain major issues regarding the panel size and the range of alterations that can be detected, CNV and translocations. Probably Pierre is going to talk a little bit about that after me. And this also poses a major issue on cost and sustainability for the value-based healthcare systems that we have in most European nations. And that also means that the size of the target population and the quality of access is compromised. And we strongly favor the implementation of a local model over the implementation of a centralized model that is the dominant one for large panels at the moment. And with this, I'm done. Thanks a lot.
Hello, my name is Pierre Sujobert. I am a molecular biologist working in the hematology lab from the Hospice Civil de Lyon in France. And today I will present to you the technical challenges of the OncNGS proposal, which can be summarized as the following goal, bringing together what has been separated. As you know, cancer genome is not so easy to study, especially because of spatial heterogeneity, meaning that depending on the site that you study, you will not find the same molecular abnormalities. Moreover, cancer genome is not the same during time because of Darwinian evolution of cancers during time. And we really need to monitor treatment response to detect residual disease and to detect as early as possible relapses in order to propose preemptive treatment when available before the patients develop clinical signs and symptoms due to relapse. So basically, if you compare the advantages of solid and liquid biopsies, solid biopsies obviously will remain very important because they are the only one to get histological analysis of tissue, and this is the gold standard of diagnosis of cancer. However, for all the other features, liquid biopsies seems to be very interesting to resolve spatial heterogeneity, to identify targets. The material is very easily available due to simple blood sampling. You can repeat this measure as much as you want. And this is very easy to standardize, probably more than for histological analysis. However, there are a lot of technical challenges before liquid biopsy can become the standard of treatment of cancer. First, we need to optimize the pre-analytics, meaning that we need to obtain very good quality ctDNA. It means that we have to make some choice, such as what to sample, blood sample or other fluids, at what time of the treatment or at what time of the day we have to sample the patients. And there are specific pre-analytical constraints on ctDNA analysis. We need absolutely to avoid white blood cell lysis in order to avoid contamination of cell-free DNA by this DNA. So you need to choose the good sampling tube, to choose the good transportation system. You need to separate as fast as possible the plasma from the blood cells. ctDNA extraction, quantification, and conservation are also specific points that need to be optimized in order to optimize the analysis of ctDNA. The goal of the OncNGS PCP is to develop the best solution for the analytic part of liquid biopsies. It means that all these features have to be optimized. We want a panel which is as much informative as possible, which means that for all patients tested, we want to find a marker to help for the diagnosis of cancer. We need a sensitive solution able to detect variants with very low allelic frequency. The solution has to be reproducible, robust. We need as limited wet lab handling as possible. The bioinformatics should be standardized as well as the clinical report. In order to be clinically useful, the time to result should be less than 10 days because the clinicians will expect the result to decipher what treatment they will give to their patients. And in order to be clinically useful for all the citizens of the European community, the price has to be reasonable in order to make it accessible for all citizens. Finally, flexibility is important because the solution should fit for one sample and also for many samples, and it should also be flexible to adapt the panel to scientific evolutions. So our principal research and development needs are the following. We need a state-of-the-art panel design with maximal informativity, with all the clinically relevant targets regarding the diagnostic, the predictive and prognostic biomarkers, and the biomarkers of theranostic implication. The price, once again, has to be in line with standard of care applications. The wet lab should be automatizable with internal quality control and a limited time on hand. And bioinformatics should be standardized, open access, with data interoperability, databases reconstruction, and all, of course, should be compliant with the GDPR regulation of the European Community. There is a huge market opportunity regarding ctDNA analysis because it will become the standard of care for the analysis of the molecular landscape of advanced cancers and lymphomas. We want to develop personalized medicine, meaning that for each cancer we will have one molecular analysis in order to decipher what treatment we should give to the patient. And finally, ctDNA will be very important for the longitudinal monitoring of cancer patients in order to follow the treatment response and the relapses as early as possible. So I thank you very much for your attention, and will be available for the question and answer session if you have some point to discuss with me. Thank you very much.
Okay, thank you. And now we have a presentation from Rosanna on the process of the PCP. So Rosanna, the floor is yours.
Yes, thank you, Mark. Thank you very much. Next slide. So, well, I'm Rosanna Sandrello from Aquas, as Mark introduced me already, from the Agency of Health Quality Assessment of Catalonia. And now I'm going to introduce you to the pre-commercial procurement procedure of OncNGS. This, just to mention, that I think is very relevant now to highlight that the presentation from Mark, from Luca, and well, the three presentations from the previous speakers are approximately the same that we did a few months ago. And this is very relevant because the preparation phase, in fact, is still valid for OncNGS. And now we are launching this pre-commercial procurement procedure based on preparation we did already since the beginning of the project. This means that the ambition and the object of the contract of our buyers is all the same. What now we are modifying is adding somehow some flexibility, as Mark was explaining, in a way that the industry can decide by themselves, how they can align their business models to the business case of the buyers, to see to how then the relationship in the future will be fruitful for the both parties. That's the key, and this is what really for us is very important now to highlight now, so that for the industry is meaningful because they can they have more now room to decide how they can align their business model to buyer's business case, but the needs are always the same. And once we will launch the pre-commercial procurement tender, this is an open call, is an open call to all the economic operators with experience in the field, and from there they will be selected depending clearly on the availability of the budget for phase one. But by the way, we assume that they will be selected 4 bidders to to run the phase one, that is consisting in solution design. This phase one contract, so the solution design contract, will last 4 months. And at the same time that we enter into contract of signing the phase one contract, also the contractors will sign a framework agreement. A framework agreement that will be valid across all the competitive phase of the pre-commercial procurement. It's important also to highlight that when we are talking about pre-commercial procurement, we are meaning that we are contracting research and development services. So there will be a phase one where the contractor will be requested to perform, to execute a solution design. And after the end of the contract, they will be evaluated. They will be evaluated, monitored along all the contract. And if their solution, their deliverable, their milestones, and what they're proposing is promising, and is evaluated like feasible, to then one day to be implemented, deployed in the operational context, then they will be invited, to submit a bid to phase two. So if we will pass from this bidder phase, the open phase of the open call, to be a contractor phase one, and to be a bidder again during the call off to enter in phase two. Then the offers will be evaluated, and the best bids will be awarded the contract, phase two contract. We imagine that there will be maximum 3 bidders, 3 contractors for this phase two. In any case, again, this will also depend on the different, we have a fixed volume for phase two. So this is, again, we depend on the bids that we will receive. And from there, during this phase, the contractors will be invited, to prototype and to go through analytical testing. Again, this phase will last 12 months. Here the budget per contractor is maximum around 700,000 euros, and during the monitoring of the contract and assessing then at the end of the contract, if the contractors are successful, they will be eligible to submit an offer for phase three. So there will be another call-off, and with the idea that in phase three, we will have at least 2 contractors. That these 2 contractors will go through the clinical validation of a limited set of pre-commercial solutions at the pilot site. And the duration of this phase will be 15 months. Here per contractor, the maximum volume is 2,100,000 euros, approx. And then at the end of this PCP, we will have, if the buyers are satisfied with this clinical validation and at the pilot sites, we will have a completely new procedure, so completely separated by this pre-commercial phase, that will be a public procurement of innovation. So we have a clear separation of the R&D phase from the public procurement of the solutions. And please, Barthelemy, can you move to the next slide? That's important also to highlight that during this procedure, we will follow clearly the public procurement principle and the principle of a treaty on the functioning of the European Union. Now that we will be all the process, all the procurement process, and all the procedure along also the different contracts, phase one, phase two, phase three, will be transparent, equal treatment, no discrimination. All suppliers, providers will be offered with the same condition, and no discrimination, mutual recognition. We will recognize all the certificates from the different issued by the different authorities of the different EU countries. Open competition. So it's open to all qualified organization individuals, proportionals. All the requirements and conditions will be reasonable in proportional to the object of procurement, and also sound financial management. The contracts will be are designed, awarded, and monitored in line with the principle of economy, efficiency, effectiveness. Next slide, please, Barthelemy. Clearly, also at the same time, since we are co-funded, this procedure is co-funded by the Horizon 220, we will need also to follow Horizon 220 rules. Next slide. Meaning that for Horizon 220 gives a specific definition of pre-commercial procurement in compliance, by the way, by the European Commission communication, that what do we mean with procurement of R&D services. So we need to cover activities such as solution exploration, design, prototyping, to the original, up to original development of limited volume of first products or services. We are not procuring already off-the-shelf solution. We are not procuring something that already exists. So we are procuring research and development services. And also we are procuring this service, making this procurement, sharing benefit and risk under market condition. So the buyers will share with the suppliers at market price the benefits and risk related to the IPRs resulting from this research and development. This also procedure is designed in competitive phases. And in fact, we already say that we are going through phase 1, phase 2, phase 3. The solution design, prototyping, analytical testing, and validation at the critical site, the pilot site. Meaning also that there are several suppliers that will provide the same services for each of the phase, and that the R&D is split in phases. And also that we are completely separating the procurement of R&D services from the deployment of the commercial volume of end products. Next slide, please. Since we are sharing risk, the buyers are sharing risk and benefits with the suppliers, the procurers do not reserve the R&D results exclusively for their own use. This means that the R&D provider generating the results in the PCP must own the attached IPR. The suppliers at the end will own the attached IPRs. And the procurers must enjoy royalty-free access rights to use the R&D results for their own use. And additionally, the R&D providers also need to commit to commercially exploit the results, because this investment that the buyers are doing is to give an answer to their unmet needs. So one day they want to buy it. If the R&D providers fails to commercially exploit the results, there is a clause, a provision called callback provision, that specifies that within a given period of after the PCP, the results must transfer any ownership of the results to the procurers. Only in case the R&D provider fails to commercially exploit the results, because ideally what the buyers are doing is making this effort to ensure that this at the end they can have an option to really make to procure the commercial volume of the solutions fruits of this PCP. Next slide, please. So the PCP will procure the R&D services at market price. But since at the end, and also through a transparent and competitive process, but since at the end the IPR ownership is attached to the results, is given to the contractors, in return, that's very important because this is something that will imply for you how to present, how to understand, to submit an offer. Since at the end the IPR attached are owned by the contractor, in return the tender price must contain a financial compensation for keeping the PR ownership, compared to the case where the IPR is transferred to the procurers. In this way, you are the suppliers and the buyers are sharing the risk and the benefits from of this procedure. So the procurers must receive the rights to use their R&D results for internal use, and licensing rights subject to certain conditions. And in the offers and economical offers, since the PCP is procured at market price, each co-contractor at the end will need to set the financial compensation for keeping the IPR ownership, compared to the case that the IPR is transferred to the procurers. Next slide. Also, it's important that to say that we this kind of procedure is exempted to follow the EU Public Procurement Directive. In fact, the procurers do not retain all the benefits of the R&D, is exempted from the WTO Government Procurement Agreement, because this agreement does not cover R&D services and does not constitute state aid, because are open, transparent, competitive procedure with risk and benefit sharing and market price. Next slide. Bidders can be anyone, can be any type of natural and legal person, and also bidders can submit bids either individually or in association, in consortium. And in fact, it's OncNGS also is putting at availability a web page where companies can advertise themselves in case they want to create consortium and share the knowledge, share the asset that they could bring to bid together. The place of performance, just the place of performance for executing the contract is the minimum percentage that is the 51% of the overall volume of the contract will need to be executed in EU member states or Horizon 2020 associated countries. That is just related with the execution of the contracts. Companies can come from other countries outside the European Union or outside the Horizon 2020 associated countries, but at least as a minimum, the 51% of the overall volume of the contract needs to be executed in EU member states and or Horizon 2020 associated countries. Language, Belgium has different languages, but in any case, English is guaranteed for the communication during the tenders and also the execution of the contracts. Next slide. After the PCP, if the buyers are satisfied with the validation, with the testing that is carried out at the pilot site, buyers are not obliged, but really this investment they are doing now, this effort they are doing now, is to one day then move on with the post-OncNGS procedure and so procure the commercial solutions fruits from this PCP. And this will imply a new prior information notice, a new market consultation, a new everything. And this will be very important milestones that the buyers are aiming for. Thank you.
Thank you very much, Rosanna, to lead us through this complex process with all the site and the legal aspects of PCPs. And I see that you're already reaching out to the future with the PPIs, but I hope we can get started with this thing now in real life. Yes. I was watching a little bit the chats, so I didn't really see any questions. I open the floor for questions, but of course, you are always welcome to send in questions through the chat box at the OncNGS site, and then we will look at that.
We have one question.
You have a question because I don't see it.
It is, you have a question that says, a general question, the initial tender was not successful. Minor changes have been introduced for the new version of for the tender. Do you envisage that the new tender will be successful?
Well, yes, we envisage that. Otherwise, I think we would not have launched it. And I think it's really, let's say, the general aims remain the same, but we reduced, let's say, the must-have, the obligatory things, and also we increased a lot of flexibility how you can answer to the way you give an answer, you provide a solution for OncNGS. So I think it should be possible now for more. Well, we learned probably from the market that what we demand is at this point in time either not really realistic or maybe not as mature as we would like to be. So that, yes, we think that it's more that what we ask now should be addressable by the market today. And yes. We also made an assessment amongst us because it's a long process already. It's already 4 years that I think we're working together as a consortium on this project, and still we feel that the unmet need has not been met yet. And Rosanna, you wanted to complement what I'm saying?
Yes, I think that it's important that again, because as Mark was saying, the level of ambition was also somehow maybe, and this is something maybe we didn't recognize with the first call for tender, was also forcing an alignment of the business model to the business case of the buyers, made in one way that was too restricting for the industry. But now the level of ambition from the perspective of unmet needs is exactly the same. It's just that we are not forcing the alignment as the buyer's perspective, but taking into consideration also that the industry has freedom to align their business model in different ways. And this is, I think, is we think as a consortium is the strengthened part now.
Yeah. And I see that there is somebody asking when we will formally launch the tender. So we hope to launch it next week through the TED platform. And then our experience is that it takes a couple of days before it's really opened. So it should be in the next coming 10 days. I say that it should be available to you again. We also will, we're in the process for the frequently asked questions, because I think we have already 52 questions being addressed on the first tender. We are modifying now a little bit according to the adaptations that we made, so that it's clear for the people from the market what is now the answers. For the rest,
you have another question, Mark. Currently, entity-specific panels are being developed in oncology study groups of larger centers for cost efficiency reasons. How do these new developments match to your expectations?
Wait, I didn't catch the first thing, the first phrase.
Currently, entity-specific panels are being developed in oncology study groups of larger centers for cost efficiency reasons. How do these new developments match to your expectations?
I think if let's say the reason that we made such a large consortium is that the cost driving, and because you see that the entity specific, I think you mean by this tumor specific panels probably. It's indeed, if you work by centers, you're related to the scope of your center, which means that the cost is mainly driven by the number of tests that you can run in a certain period. Now, with OncNGS, we envisage really a large market. So the cost will be lowered due to the volume that OncNGS is going to be used. And volume is still probably in this world the major cost driver. And also, I think we aim to create networks, and the larger centers, of course, they have quite some volumes, but even the fact that it's pan-cancer, but the modularity that we allowed is that allows centers, because I know that some centers are more specialized in particular things and not in others, to have a certain modularity in what exactly is being used. So I think, yes, OncNGS is in fact already taking into considerations these new developments. If the tender will be published next week, which is the estimated deadline for semester? About 90 days, 3 months later. So it should be somewhere, if I calculate well, beginning October. First, well, let's say around 7 or 10 October, something like that. Mid-October, say. Okay, I don't see any more questions. So anyway, feel free. Is the first code that was I think there was, Rosanna, there's still a question on the 51%, let's say, activities that needs to be done in Europe.
Yes, it's just activity related with the execution of the contract, not activity on the company in general. But yes,
and I think, Rosanna, it encompasses all the costs, personnel, materials, as long as it's done here in Europe.
Exactly. The volume of the contract in general, that is, this is research and development contract, and you will need to demonstrate that more than 50% of the contract is research and development, because it's not a supply contract. On the other side, as Mark is specifying, the 51% needs to be carried out in Europe, the volume in general. So it encompasses everything.
Yeah. Okay.
And also, I didn't specify a bidder can subcontract, and also the subcontract maximum is 50%.
I see how in this call the IVD certification is relevant. Well, I think we do not aim that you can really get IVD certification with the work that is done here, but it should be taken into consideration because in the future, OncNGS will be subjected to the IVD. So it's going to be also subjected probably to the medical devices because it has a decision system included in there. So this should be at least addressed, but that is one of the requirements in the call for tender that you will be able to read. But it's not the idea that you demonstrate the certificate for the registration.
And then Barthélemy is reminding us something that already also mentioned, but is very important, that this is very complex contract, and so also the composition of consortia to bid together with other companies is something allowed, is something good if one company cannot do it everything by its own, and we have in the OncNGS webpage, we have a matchmaking page, specific matchmaking, find a partner. And so if someone has, again, some already some technology, some experience they want to share with others to create consortia, this is, we think that is a good way, no? To
Yeah. Okay. I don't see any questions anymore now. So anyway, you still have the opportunity to ask and address questions through the chat. And I think I now would like to close the session. I would like to thank you, all of you, for your participation and active discussions on the project. And I'm convinced that the OncNGS PCP is something interesting for the market. We as a consortium, and I should also say our supporting entities, are still very keen on starting to work on it. We fully believe that this would be a major step forward for the benefit of better treatments for patients. And I'm eagerly waiting for the offers that we may receive, let's say by the end of September, early October, on our call for tender. So I would like to thank once more Rosanna for the nice presentation, and Luca and Pierre for their contributions, Barthélemy for helping us with the slides, and I wish you all a good evening. Bye-bye.
Bye. Thank you. Thank you. Bye-bye.
Good evening.